Hello. Today we see about extracellular enzymes. First, let's see what is extracellular enzyme. It is also always called exozyme. It is an enzyme that is secreted by a cell and functions outside that cell. So these are called exozyme. Exozyme or extracellular enzymes are produced by both prokaryotic and eukaryotic cells, have been shown to be a crucial component of many biological processes. These enzymes are very important for the many biological processes. Microbes can take up nutrients of high molecular weight, can able to take a low molecular weight, including sugars, amino acids, but also oligopeptides, nucleosides, and small organic phosphates such as glycerophosphate, etc. This type of substances is not digested by the microbes. For this digestion process, various bacteria, fungi, and protozoa produces variety of extracellular enzymes. So, many high molecular, low molecular weight substances cannot be degraded by the microorganisms for the nutrients or the virulence factor for product themselves. They also produces some enzymes. So, the enzymes may act as the pathogen or may act as for the nutrients for degrading the compound to further getting energy. These enzymes often play a role in disease by attacking tissue constituents. How the enzyme may be function of cells acts on attacking the tissue constituents. For example of this type of enzymes are Hyaluronidase, Hyaluronidase, phospholipase, Chitinase, Collagenase, Fibrinolysin or Fibrinokinase enzyme. So these are the examples of extracellular enzyme acts as a pathogens. That is acts as a virulence factor. So these 6 enzymes are act as a virulence factor. These enzymes are produced by microorganisms, also interfere with the normal mammalian functions. So this will interfere with our normal functions. Some of these enzymes contribute to the virulence of microbial pathogens. They are phospholipase, Chitinase, Fibrinolysin or kinase enzyme, Collagenase, Hyaluronidase, Collagenase. Next is about one by one of the enzymes. First is about phospholipase. This enzyme is produced by the microorganism Staphylococcus aureus. So it is produced by the Staphylococcus aureus. The main function of the enzyme is destroying the animal cell membrane. This enzyme can also act as a chemolysis, that is causing the lysis of red blood cells. So it is produced by the microorganisms Staphylococcus. The main function of the enzyme is destroying the animal cell membrane. This enzyme can also act as a chemolysis, that is a breakdown of blood, and it also acts as a lysis of red blood cells. Second one is Chitinase. This enzyme is otherwise called as a toxic enzyme. For example, toxin of Clostridium perfringens is a Chitinase enzyme. This enzyme hydrolyzes the lecithin, which is a lipid component of eukaryotic membrane. So it cleaves the membrane by hydrolyzing the lecithin. The hydrolysis of lecithin by the alpha toxin of Clostridium perfringens. It destroys the many cells and it's a cause of extensive tissue damage in the disease. So it will cause the damage. Chitinase also acts as a chemolysis, causing lysis of RBC in addition to the destroying cells of various other diseases. In addition to the destruction of other diseases, this enzyme also breaks the RBC cells. So RBC is very essential for the mammalian. Next is the Fibrinolysin or kinase enzyme. Some Staphylococcus and Streptococcus species produces Fibrinolysin. Fibrinolytic enzymes are Streptokinase and Staphylococcus. It is named based on the microorganism. If it is a Staphylococcus produces, the enzyme is Streptokinase. If it is Streptococcus, it produces the enzyme is Streptokinase. They catalyze the lysis of clot. These two enzymes break down the clot. So when the clot will occur in a microorganism, it enters into our body. Our immune system captures the microorganisms by forming a clot. So this microorganism escape from the clot, they produces this fibrinolytic enzymes and break the clotting. Clot occurs around the microorganism. It will be escape and will enter into our body. So the action of these two fibrinolytic enzymes may enhance the investigation of pathogenic strains of Staphylococcus and Streptococcus by preventing the fibrin in the coast from walking on the area of bacterial infection without the action of fibrin. That is, it can easily move through the infection site, it will multiply. Without the, it breaks the action of fibrin, which is our immune system action. The pathogens are free to spread to the surrounding area. Whenever the clotting occurs, it will be break the, using this enzyme, they break it and will be move out. So this is action of fibrinolysin or kinase. Next is the coagulase. This enzyme is produced by the microorganism Staphylococcus aureus. This enzyme converts fibrinogen to fibrin. So in previous section, they found that when our immune system make a clot and capture and then by its phagocytosis mechanism, it will be escape. But in coagulase enzyme, here the microorganism produces the coagulase enzyme, it will protect itself by forming a clot. So it will be secured within the clot. It will be escape from the our immune system action. The enzymes they convert the fibrinogen into fibrin. They form a clot. The Staphylococcus aureus species produce this enzyme and of fibrin around the Staphylococcus cells, presumably protects these cells against the circulatory defense mechanism of the host. So it will protect themselves from the circulatory defense mechanism of the host. Hyaluronidase. This enzyme is produced by the microorganism is Staphylococcus biogen. The main function of this enzyme is to break down hyaluronic acid, the substance that holds together the cells of connective tissue. Pathogen that produces, so in our all joints, the very important for any compound is hyaluronic acid. When the microorganism produces hyaluronidase, it breaks the hyaluronic acid. So it is referred as the spreading factor. Various species of Streptococcus, Staphylococcus and Clostridium species produces hyaluronidase. So when it is a breakdown this hyaluronic acid, so we have a connective tissue will be breaks. So it will easily enters into our cells. It will produce multiply and it will infect us. Next, collagenase. This enzyme is secreted by the bacteria is Staphylococcus aureus, Clostridium perfringens. So it is also similar to the hyaluronidase. Here the main function of this enzyme is to break down the proteins of collagen tissues. So collagen is also very essential for moving our joints. So when the collagen will be break down, pathogen will be easily enters into our system. So the breakdown of fibrous tissues enhances the invagination. Invagination enters the pathogen microorganism into the host. So this is collagenase. So these are the 6 important microorganisms that give virulence of pathogens by enhancing the ability mechanism to proliferate within the body tissues or by interfering with the normal defense mechanism of the host organism. So these are the some microbial enzymes. They contribute to the virulence of pathogens by enhancing the ability of the microorganisms to proliferate within the tissue. So some enzymes will access the pathogen to enhance the microorganisms to enter into the body. Some enzymes will interfere with our normal defense mechanism of the host organism to protect the microorganism. So two functions will be done by this microorganism. So one, it will be protecting microorganisms against our defense mechanism. Second one, it breaks our defense mechanisms and produces virulence to the host. So these are the functions done by the microorganisms. Next we see about some of the type of extracellular enzymes are secreted for the other purposes, for nutrient breakdown of substances into small ones for their growth. Many polymeric substances such as cellulose, chitin, starch and xylem pectin are too large to transport through the pores of the cytoplasmic membrane. One strategy employed by the microbes is to use a substrate in the extracellular enzyme. They have to use the substrate by using the extracellular enzyme, break it into smaller one. This enzyme degrades the polymeric substances into small units and it can be transported across the cell membrane and metabolized. Besides the role in converting impermeable substances into milk. So, the function is they converted into large molecule into small molecule. Another one is it make it the impermeable substances into permeable substrate. Example of this type of extracellular enzymes are amylase, cellulase, it's a cellulase, not a cellulose, it's a cellulase, pectinase, chitinase and silanase. So first we see about amylase. Alpha amylase are produced by fungi and bacteria. Its ability to degrade starch by producing amylotic enzyme is called amylase. Many soil fungi are active producers of amylases. Amylases are produced commercially by the use of Aspergillus oryzae, Aspergillus niger. Among bacteria, the bacilli and several streptomyces are the active producers of alpha amylase. This alpha amylase enzymes are mainly attack the alpha-1,4 beta alpha-1,4 glycosidic bond, produce maltose and oligomers with 3 to 7 glucose residues. So alpha amylase break down the large molecule starch into maltose and the oligomers. In starch, alpha-1,6 linkages are mainly cleaved by the amylo-1,6 glucosidase. It ensures the cleavage of branch points. It also converted to maltose and then as maltose is converted into oligomer, it will be transported into the cells and cleaved phosphoryctically. So, the amylase enzyme is produced by fungi and bacteria. So, mostly the fungi are the Aspergillus oryzae and Aspergillus niger. In bacteria, bacilli and streptomyces are the active producers of alpha amylases. It can able to break alpha-1,4 and also 1,6 linkages. Anything will be converted into maltose and the oligomers. It will be transported. That's about the amylase. Next is a pectinase. Many bacteria and fungi have the capacity to metabolize pectin. The pathogenicity to plants of some microorganisms is due to the ability to excrete pectin dissolving enzymes. Mainly this enzyme is produced by the bacteria is Bacillus polymaxima, Clostridium pectiniorum. So this is about the pectinase. Second, chitinase. Streptomyces criceus secrete two types of enzyme, chitinase, chitobiase. This enzyme degrades the chitin. This degradation apparently occurs by the simultaneous attack of chitinase at many points along the polymer, yielding the N-acetyl glucosamine and chitobiose and chitodiose as the product. So this microorganism produces the two enzymes, chitinase and chitobiase. Chitinase break down the chitin. It produces the L-acetyl glucosamine, chitobiose and tribiose means two N-acetyl glucosamine. N-acetyl glucosamine is a monomer. It is a polymer. Chitin is the polymer of N-acetyl glucosamine. So it produces chitobiose and chitodiose. So next another one enzyme, chitobiase. It degrades the chitobiose into N-acetyl glucosamine. Likewise, other will be converted. So this is about the chitinase. Next is the silanase. The main function of this enzyme is to degrade silane to cellulose and cellulodiose. So similar like that, large fragment of silane is converted into cellulose and cellulodiose. So it is produced by the enzyme silanase. It is degraded by the enzyme silanase. In bacteria, silanase is formed constitutive in some organisms like Clostridium species. So silanase enzyme is mostly produced by the microorganism Clostridium species. It degrades the silane into cellulose and cellulodiose. Next one is cellulase. The main function of the enzyme is to degrade cellulose. It is produced by the microorganism Sphaerococcus species. Most of the Sphaerococcus species produces cellulase. So come to the conclusion that extracellular enzyme secretion is very essential for the survival of the microorganism because this enzyme can be used both for obtaining nutrition and also production. So enzyme is very essential for the microorganism survival. Okay. So this is the reference. Thank you.